So, good afternoon, one and all. Welcome to the week 6 live discussion on BioMEMS and Microfluidics. So, the course instructor for this course is Dr. Shanthanu Bhattacharya, and the live session is handled by me, Sabareesh. And I am the PhD scholar and the PMR of in the Department of Mechanical Engineering at IIT Palakkad. And my research area is related to passive cooling applications. So, the objectives of this 1 hour live discussion, as you know, is a glance through the weekly video sessions. Then comes the Q&A or the problem solving. Then the doubts or queries you have maybe cleared through this live sessions. So in the last week, the following topics were covered. One was surface electrophoresis, then protein polymerization, transcription and translation processes. So we have previously seen capillary electrophoresis in the previous week lectures, which includes the general gel electrophoresis types. So this is the surface electrophoresis type. Then comes the protein polymerization, the creation of the polymers from the protein particles. Then the transcription process, the translation process, which are all in the part of the synthesis of the DNA or the analysis processes. Also, the transcription and the translation in bacterial cells were also discussed. Then, and the different types of protein complexes and its use in the diagnostics or the therapeutics is also discussed in the last week lecture. I hope all of you have gone through the lecture. So one of the important aspects that has been discussed in the last week was the DNA Coulter counter. And in this, this is actually a device to monitor the system analysis. So DNA counter is a particle analyzer in which the pair of electrodes actually detects the effect of passing particle on the impedance. So impedance is denoted by the letter, or we know that it's a Z. Z is actually a ratio showing the ratio of the resistances, the capacitance of and inductance in a circuit. So impedance is the total resistance in a circuit. For a suppose we have a RLC circuit. So from that we know that it's a second order system. So impedance refers to the total resistance of the system under analysis. So we know that they will create a phase shift and difference between each other when they are connected in series or parallel. So the effect of a passing particle on these impedance is studied with the help of a DNA Coulter counter. We know the other device like Geiger counters also, which measures the number of particles passing through a point at a particular point of time. Similar to that, this DNA Coulter counter is also another kind of particle analyzer system. But the difference is that this Coulter counter will be used to measure the total cell count in a microbiological sample. Suppose we have a kind of microbiological sample containing different types of maybe bacteria or another kind of substrate to be analyzed. And if they are mixed together, the counter is able to detect the particles that are present in this substance separately. So the technology that it uses is by means of an electrical circuit. So initially, this process was developed to count the blood cells quickly by measuring the changes in the electrical conductance. So these cells will be suspended in a conductive fluid. Suppose we have a different types of cells, maybe bacterial cells or another kind of cells. We are going to suspend all these particles in a particular conductive solution. And these cells will be suspended here as shown here, and then the conductive fluid will be allowed to pass through a small orifice. So there will be a particular rate at which this fluid will be passing through it, and this fluid contains our particular cells to be analyzed. So that will be taking these particles along with that, and the measure or the count can be conveniently measured with the help of this DNA counter. So you can see in this image here, there is a volumetric metering device, there is a valve, and there is an aperture through which the fluid may enter and enters the system, and you can see the magnified version here. So there is a counter start stop switch or a button here so that it will take starting the countings as soon as we switch on the system. Then there is a stirred suspension of particles in electrolyte solution. You can see here this is stirred suspension particles in the electrolyte solution, and there are two electrodes, plus and minus, and as soon as the fluctuation happens in this circuit or in this fluid, that will be recorded by this electrical circuit, and they will be converted or they will be transmitted into a pulse amplifier. So pulse amplifier, what does it do is the signals which are coming from this system is very weak, which means it will be in the several range of millivolt. So if some kind of noise is interfering with this, what it will do is it will disturb the input signal. So in order to avoid that, we will be using a pulse amplifier to amplify the signal. So the signal will be amplified with the pulse amplifier, and it will be converted into several volts. So after that volts, it will be converted into several range of 1000 or say 1000 millivolt or so, and this signal will be separately fed into, there is an oscilloscope pulse display which displays the pulse detected. There is a counting circuit, and there is a pulse height analyzer. So how much is the height or the rapidity of the, or the rapid movement of the particles or the cells, that will be determined by the pulse height. And finally, taking all this, the output signal will be displayed. So that is a principle of a DNA counter. So they are essentially using the DNA analysis type. So I was previously saying about the Geiger counter also. So Geiger counter is actually an electronic instrument used for detecting and measuring the ionization radiation. detection of the ionization radiation will be done by the Geiger counter. So they are particularly using radiation dosimetry, radiological protections, experimental physics, and the nuclear industry. So that is what a Geiger counter is. So they are especially used in the nuclear industry to detect the radiation frequency or the radiation level. Next topic is actually one of the important topic discussed was secondary beta strands. So a beta strand is an element of a secondary structure in which the protein chain is almost or nearly linear. So it is separated from a protein chain, and that protein chain is actually linear in structure. So what is the importance of this beta strands is that it can hydrogen bond, HH bond can be formed to form a beta sheet. Also, it is referred to as the beta pleated sheet. You can notice as shown in this figure that the participating beta strands are not continuous in the primary sequence, and they do not even have to be close to each other in the sequence. That is, the strands forming a beta sheet can be separated in their primary structure by with the help of a long sequence of amino acids that are not the part of the sheet. So beta strands are not continuous in the primary sequence, and they do not have to be close to each other in the other sequence also. And the strands can be separated by means of amino acid groups. So please note that amino acid groups can separate these primary structures. I hope it's clear. Then approximately a quarter of all residues in a typical protein are beta strands. So in a typical protein, quarter of all the residues are generally beta strands, even though they may vary with the different kinds of protein. So in figure 2, you can see a beta strands that are actually parallel. So 1 type, 2, 3. So you can see how they are arranged. The figure shown is a parallel type. See, they are connected in this manner, separated. The protein chain is nearly linear. That can be observed like this. So they are nearly linear. Next comes the parallel beta sheet. So the different types of secondary beta sheet is the parallel that we have shown here. So all the bonded strands have same N to C direction. And as a result, they have to be separated by a long sequence stretches. And also you can see that the hydrogen bonds, the HH bonds are equally distanced, or they are spaced equally to each other. So in figure 3, you can see the anti-parallel beta sheet, which is the next type. So what they do is they run in alternating directions. So therefore they can be quite close on their primary sequence. And the distance, you can see the distance between successive hydrogen bonds are not constant. And they alternate between shorter or longer. So the distance, you can see the distance is short. The distance may be long, short, long, short, long. And the arrangement you can see here. It's an anti-parallel arrangement. See, this is one in the top direction and this is in the opposite. Anti-parallel, there is a 180-degree shift between their arrangement. The third type is the mixed beta sheet. So this is actually a mixture of parallel plus anti-parallel hydrogen bonding. So nearly about 20% of all the beta sheets are of the mixer type. Okay. So another topic I would like to introduce to is the term codon. Before that, we know what is a genetic code is. So actually, genetic code is the set of rules for any living cell. So this actually the genetic code carries the information of the genetic material. So in any living cell, the genetic material will be stored in the form of a particular genetic code. So what it does is, so codon is a specific sequence of 3 consecutive nucleotides that is a part of this genetic code, and which refers to a particular amino acid in a protein. So what it determines is there is a particular code or codon. It's an arrangement of the consecutive 3 kinds of nucleotides, maybe GCU, AUG, GCC, any type. So it's an arrangement of 3 consecutive kind of nucleotides, and what it does is it tells the material or the cell how to process or synthesize a particular kind of amino acid in a protein. So it can tell a particular cell whether to start or stop protein synthesis. So accordingly, it will be monitoring the genetic information. So there's a particular pattern in which any kind of a substance will be synthesizing this proteins. So all the genetic materials in this, whether it be hair, color, or anything, it's actually a consequence of this codon arrangement. So there's a particular pattern in which these codons are synthesizing the proteins. So which is unique for a particular living being or a particular cell. So there's a particular pattern of synthesis which determines the overall behavior of the cells. So if we look in a DNA like this, so suppose there will be it's an open standard whether it be, so DNA is the information carrier of a cell. So if we look at that, there will be a particular arrangement GCU, which is actually a codon 1. Then comes maybe ACG, which will be a codon 2 type. GAC may be codon 3 type. So all these things are telling the cell to do the protein synthesis by influencing the amino acid. I hope it is clear. So please note that these codons are generally 3-letter. There's a 3-letter nature of these codons. So the 4 nucleotides present in mRNA, which includes the A, U, G, and C. So totally they can produce a combination of 64. And out of the 64, 61 will be representing the amino acids. And the remaining 3 will be the control signals or the stop signals, which trigger or end the protein synthesis, That is what a codon means? So next, one of the important concept that has been discussed last week was the Michaelis-Menten equation. So why is it used? Like why is it important? Actually, they have been used widely for over a century to estimate the enzyme kinetic parameters from the reaction progress curves. So, the reaction, how does it progress over a time? So the rate will be increasing and after that it will stabilize, maybe in the case of an enzyme. So in the reaction progress curve, which is also called as the progress curve assay. Progress curve assay. So this equation is used to determine the enzyme kinetic parameters from the reaction progress curve of the substrate. So this equation assumes, or there is an assumption for this, that is, there is a rapid reversible formation of a complex between an enzyme and its substrate. So if there is an enzyme and a substrate, there is a rapid reversible formation of a complex between the substance and the substrate. We know that substrate is a substance upon which it acts to form a product. Yeah. So it also assumes that the rate of formation of a product P is proportional to the concentration of a complex. Suppose if the concentration of a complex is C, it assumes that the rate of formation of a product P is proportional to the concentration of the product. So dP by dt is proportional to concentration of C. So in this equation also, it has velocity of the reaction is greatest. From the observation, we find that when all the site at which the catalytic activity can take place on the enzyme, the molecules are filled with the substrate also. That is when the substrate concentration is very high. These relationships provides the basis for all kinetic studies of enzymes and also have been applied to the investigation of the effect of the carriers upon the transport of the substances through cell membranes. So the relationship is actually providing the basis for all kinetic studies or the behavior of the enzymes in different substrate. So the equation is telling us how that is getting influenced by the substrates. So the equation is V0 equal to V max of S by Km plus S, where V0 is the initial reaction velocity. Vmax of S is the maximum velocity. Km is the Michaelis constant. And S is the substrate concentration. So that is actually the Michaelis-Menten equation. So you should understand why this Michaelis-Menten equation is important. It is essentially important because it is important for understanding the kinetics of the enzymes that are used in cellular synthesis. So there is a cellular synthesis happening in every enzymatic reactions. So the cellular synthesis will be determined with this uh, presence of the enzymes, and that will be measured by using the reaction velocity using Michaelis-Menten equation. Okay. So there is also an importance for this Michaelis constant. So this is actually a value which is equal to the substrate concentration state, which is S dash, at which half of the enzyme active site, or the sites in which this enzyme comes into action, are saturated with the substrate. So the saturation state will be determined by the Michaelis constant when the half of the enzyme active sites are saturated with the substrate. So you can also understand you should understand the concentration of each of the substances present in the graph, or the concentration of each of the substances present in the chemical reaction. So if you plot concentration on the x-axis, y-axis, and time on the x-axis, time of the reaction or any reaction happening. So the rate of the reaction, we are denoting it. So that will be a rising curve. This is the P, then the substrate concentration. That will be decreasing over time. Next comes the enzyme concentration. Then comes the complex enzyme substrate. So the enzyme will combine with the substrate to form a third substance. So if you are looking at the start from zero, and finally this will be the enzyme substrate that will be getting formed. So if you look at a simple equation, we can represent this also. So the enzyme plus substrate, when it is equilibrium, this is the forward rate of reaction and this is the reverse rate of reaction. It forms a complex substrate which combines with the substrate. So the enzyme and the substrate together forms a complex. You can see here, which will finally disintegrate or to form enzyme plus product. So this is the K catalyst, or the catalytic rate constant. This is mostly it will be a single direction curve. So that's an kindly change that. So this is a single kinetic rate of reaction forming the enzyme and product. So the enzyme is separated at the end of the reaction. We know that substrate actually acts as a binding material also. So let us go to the Q&A part. So first question, where does the translation take place? Nucleus, cytoplasm, nuclear membrane, ribosome. So translation. So what is actually the translation process? You can leave the answer in the chat box also. So please note here. So translation is a process by which there is the genetic code which is contained in the messenger RNA or the mRNA and that is getting decoded to produce a specific sequence of amino acid in a polypeptide chain. So that is a process by which the genetic code contained within the mRNA molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. So it occurs, where does it occur actually? So the options given are nucleus, cytoplasm, nuclear membrane. So these are not the options. The correct option is ribosome. So what is ribosome? So RNA or the ribosome is a complex molecule and proteins that forms a factory for protein synthesis. In cells. So the ribosomes, you should understand that it has got primarily two functions. First function is decoding the message. Then it forms the peptide bonds. So what are the four processes of translation? So the four processes of translation in DNA are activation, initiation, elongation and termination. So these are the main steps of translation process in DNA. Activation, initiation, elongation, and termination. Okay. So next question, DNA sequencing refers to which of the following? It is used as a technique to determine the sugar sequence in a DNA molecule. It's a technique used to determine the base sequence in a DNA molecule. Then technique used to determine the phosphate sequence in a DNA molecule, and all of the above. So which is the correct option? DNA sequencing is used to determine the base sequence in the bases, the 4 bases. So sequencing is connected with the base sequence analysis in a DNA molecule. So the correct option is option B, technique used to determine the base sequence in a DNA molecule. So how can we sequence DNA? Can anybody of you say how this sequencing happens? Then we can do the process of this sequencing. Yes, one option is Sanger sequencing. So what are the different methods in Sanger? So first we will create DNA sequence for chain termination PCR. Then there is size separation by gel electrophoresis. Then gel analysis and determination of DNA sequence. So please have an understanding of this DNA structure. So there will be a base, then there will be a phosphate molecule, then there will be an OH minus, then there's a sugar base. And the bases, as you may know, is adenine, cytosine, guanine, thymine, and each of the two of them forms the complementary pairs. Okay. So next question, what do we use to cut the DNA before gel electrophoresis? So options are restricted enzymes, DNA polymerase, lipase, and helicase. So which is the answer? Option A, restricted enzymes are the answer for this question. So to cut the DNA, we use the restricted enzymes before gel electrophoresis. So why is gel purification important? It allows you to isolate and purify the DNA fragments based on their size. So the procedure actually starts with an agar-gose gel electrophoresis, and then there will be separation of DNA by the difference in the length in the base pairs. Then following electrophoresis, you can cut the DNA bands out of the agar-gose gel and purify these DNA samples. So that's a simple procedure associated with this DNA gel electrophoresis. So you may know that the DNA is negatively charged. So when an electric current is applied to the gel, DNA will migrate towards the positively charged electron. So why is DNA negatively charged? Due to the presence of the phosphate ion. Okay. So you must understand these cuts are always made at specific nucleotide sequences. Okay. Okay. To the next question. How many DDNTPs are used in Sanger's method? So what is DDNTP? DDNTP means the dideoxy nucleotide. So in Sanger's method, the question is how many DDNTPs are used. So in Sanger's method, there will be 4 types. So what are the different 4 types used? One is DDATP, then DDGTP, then DDCTP, and DDGTP. So when they are incorporated into the amplification reaction, the chain will be terminated. Okay. So what happens when a DDNTP is added? You would get more sequence close to the primer and make it more difficult to read. Sometimes when the sequence is 200 to 300 nucleotides further down. So the correct answer for this question is option A, 4. Question 5, in gel electrophoresis, what does the gel act like? Is it a gate, a barrier, a filter, or none of the above? What is the answer for this? Is it a gate or is it a barrier? No, in gel electrophoresis, what it does is it is a filter. So it separates out the gel electrophoresis. What does it do? Based on the size of the protein or the RNA type, it will permit the entry of the molecules. So essentially, it's not a barrier. So it's not a wise option. It's not also a gate. The correct option is it acts as a filter. That is a function of gel in a gel electrophoresis. What is a gel used in electrophoresis? Agargose is the type used. Okay. Question 6. Which of the following sequencing method uses PCR for generating sequence template? LMPCR, then Sanger's method. Both LMPCR and Sanger's, and none of the above. So the question is, which of them uses PCR, polymerase chain reaction, for generating sequence template? So the correct option is only LMPCR. It's not applicable with Sanger's, so C's, B and C are wrong. So LMPCR is the correct option. Next question is, what are antibody molecules? So antibody molecules, what are they? They are actually the options you can see. Option A, we recognize carbohydrates via a lectin head. So that's the false option. They're also not produced by T cells. It uses the Fc region to bind. That is also not correct. So they are essentially the globular proteins found predominantly in the gamma region during electrophoresis. In electrophoresis, globular proteins are found. So if a question is asked, where is this antibody molecules are produced? Where are they? They are actually produced by the B cells. Please note that antibodies are produced by B cells, and they are producing several billions of forms, and each with a different amino acid type and a sequence and different antigen binding site. So the T cells, they are actually the fighters for the invaders, or they are produced by the cytokines. Okay. So next question. Monoclonal antibodies currently used clinically. Like what is the function of the monoclonal clonal antibodies? That's the question. So 1, 2, 3, 4. Four options are there. So question is, can protect against a wide variety of viruses and bacteria. Yes. So clonal actually is due to the cloning or the specific synthesis of the antibody. So option B is correct. Then B, can reduce the inflammation associated with rheumatoid arthritis. So antibodies, the clonal type are specifically treated. If the rheumatoid type, the antibodies are created monoclonally, then that can be definitely be used for this rheumatoid arthritis. So that option is also correct. Next option are derived from the plasma of the individuals already immune to these organisms. So are they derived from the plasma of the individual already immune to these organisms? No, they are not immune. We can't generate. So that option is wrong. Then next option is each have broad specificity for many antigenic determinants. So the specificity like antigenic determinants or the specificity is comparatively less for different antigenic bodies. So the only option that is permissible are option A and B. Both are seeming to be the correct option. Okay. Next question. The antibody class found at the highest concentration in serum is, so in blood serum. What is the antibody class found? So the antibody class type or the isotypes are IgA, IgD, IgE, IgG, and IgM. So what are these Ig? Can you say what is this Ig? Where does it come from? How does it is relate? How is it related to antibodies? Yes, Ig antibody, as I said before, is the immunoglobulin. So that is what is Ig coming from. So the variants are A, then D, E, G, and M. So in serum, the common type found is G type. That is the case with the serum type. So the correct answer is option D, IgG type. Clear? Okay. So 10th question, which of the following is responsible for the initiation of RNA polymerase activity? Options: initiation site, promoter region, sigma factor, and rho factor. So what is initiation site? So initiation site is a site on the DNA from which first RNA nucleotide is described, transcribed. So it is called the +1 state or the initiation state. So there is a promoter region, there's an initiation site, and which of the following is responsible? So the options possible are a rho factor or sigma factor, but the correct option for this is the sigma factor. So option C, sigma factor is the correct option. Next question, which of the following are non-sense codons? Please read it as non-sense. Non-sense which it does not sense, not in the literal form, the non-sensory codons. Or which is not sensible, or what is which is, you can say it is irrelevant codon type. So you need to have an idea of the codons arrangement. So as I said before, codons are genetic information carries a three-member sequence of A, U, G, and C. but there is a particular arrangement for such sequence during. So one of them is not valid. So you need to identify which of them is invalid. It's AUG, GUG, UA, or UC. The first glance you can see UA is not a, because two are coming together, and that is two adjacent. There is a GG, but in between U is coming. Then AG and UU, in between C is coming. But UAA combination type is not permissible. So that's the part of the translation type. But you please note that there are types like AUU also. But UAA type is not there. So to familiarize with that, you need to understand about the codon arrangements and the different types. So that's all with the questions today. If you have any questions or doubts in the problems of the initial part or in the weekly session, we can clear it now. So I would like to mention also the how does a segment of a gene would look like. So if it is considered in a rod format, so there will be a promoter here. Then there will be codons for enzyme. Then there will be a stop codon. Then there will be one gene. Okay. I hope the session is clear with you. If you have any doubt or questions, you can always post in the discussion forum. And if still there, you can also discuss in the upcoming classes. So that's all about the week 6 live interaction. See you all next Saturday at 3 to 4 p.m. for the live interaction session for week 7. Thank you. Thanks for your patience. Yeah, thank you.